name's Tom Howe. I'm the director of the honors program. It's really pleases me to see so many people here, so that's wonderful. Our students work really hard, and this is the culmination of that hard work, so it's, I'm grateful that you're all here to help celebrate that. So we're going to continue on with Lynn Wynn, and here to introduce Lynn is Dr. Beck.
Save your applause. You're gonna have plenty left for Lynn. Teaching and mentoring undergraduate students is tough. And one of the things that makes it tough is that you always have this feeling that your students are going to go on to do great and amazing things. But you don't know what those things are yet, because they're just undergrads. Every once in a while, though, you get lucky, and you get a student who contributes to something important as an undergrad. And that's what you're going to hear today from Lynn. Lynn has contributed to something that's actually important. And I know she's going to go on and do great and amazing things because she's already doing them. She's already telling this story. The project you're going to hear about today started more than 2 years ago. Lynn applied to and was accepted to the Summer GEMS program at CU. And she worked in Dr. Freed's lab that summer. And she apparently did well enough in that role that Dr. Freed asked her back into his lab. And since that time, she's had internships in the lab, she's had paid positions, she's volunteered in the lab, she's sort of become a staple of that lab. At some point in that process, she came to me and she says, Dr. Basham, I really want to do my honors thesis research with Dr. Freed in Dr. Freed's lab. And I said, great, then I don't have to do anything. I'll let Dr. Freed do all the hard work. But I said, but you have to pick a new project. You can't, I'm not going to let you do the stuff you've been working on for a year or more. You and Dr. Freed have to come up with something brand new for this, since it's a brand new honors research project. And Lynn was a little resistant to that at first. She didn't think that was a great idea, but she did it. She came up with something brand new. So what I wanted to tell you about her talk today is there's a whole nother set of research that she's only going to allude to, that she's not really going to talk about, that she did before she did any of this. And you should at least know that there's even more impressive stuff behind the stuff that you're going to hear today. Remember this moment. In about 15 years, phenylbutyrate is going to be the standard treatment for Parkinson's disease. I think that's a real possibility. And when that happens, you'll think back to this and you'll think, oh, I heard about that 15 years ago. I heard about that treatment when I went to Lynn's honors thesis talk. And I know Lynn. She did a little bit of that. She contributed a little bit to that. And she'll have done great things. She's done great things now. So with that, I will let Lynn tell you about phenylbutyrate and Parkinson's disease and a little bit about the FDA.
Thank you for that. That was actually really heartwarming. I'm not totally sure how to turn this on.
It'll more.
Yeah.
It's going.
You just have to vamp for a minute or so.
Tap dance. I don't think it's starting on.
It sounds like
Here,
this.
it sounds
Kind of blinking green light.
Just get off.
That's coming.
Okay. Hi. So, I'm Lynn, and my thesis is on stopping Parkinson's disease with a drug called phenylbutyrate. So, James Parkinson first described the disease in 1817, and he pretty much wrote about patients with like resting tremor, rigidity, stooped posture, shuffling gait, and it was crazy how accurate he was because he basically made these observations based off of 4 or 5 people he saw walking down the street. And to this day, we still use the symptoms that he wrote about as standards for clinical diagnosis, so like it's crazy. But for how renowned his work is now, when he published it, no one really like looked at it because there was a cholera Jean-Martin Charcot stumbled upon Parkinson's old essays and reintroduced it to the public. So Charcot was the one that coined the term Parkinson's disease. And with Charcot's reintroduction, a ton of new people wanted to research and look at Parkinson's. And because of that, about a century later, Elaine Hornikovich was able to link Parkinson's disease to dopamine deficiency. So Hornikovich was looking at postmortem Parkinson's disease patients' brains, and he was like, these brains do not have any dopamine in them, so maybe that's the cause of Parkinson's disease. So okay. So I will present from back here. So Hornikovich is really a pivotal player in this whole Parkinson's disease. So he revolutionized the way we saw Parkinson's. Before him, all of the research that was done was pretty much exploratory. So people were looking at what the disease was doing, the long-term effects, like the non-motor symptoms of Parkinson's disease. But because Hornikovich pinned down Parkinson's to dopamine deficiency, he introduced the possibility of a cure. And he himself had this theory. So dopamine is a molecule that's kind of way too big to cross the blood-brain barrier, and if you take it orally, you digest it and you kind of just like sweat and urinate it out, and it's not an effective treatment. So he had this idea, maybe we could give patients dopamine precursors, and when they take it, the body's natural enzymes will convert it to dopamine once it's crossed the blood-brain barrier. So to test this theory, Hornikovich sent a vial off to his friend Walter Berkmeyer, who was in charge of this neurology facility, and Walter Berkmeyer just gave out dopamine precursors to his patients, to his Parkinson's patients. And this is totally unethical. If you do this today, yeah, it was not a good thing. So if you do this today, you'll probably get kicked out of your institution. But at the time, the FDA wasn't a thing, drug regulations weren't a thing, so here Walter Berkmeyer is just giving out dopamine precursors. And it's kind of a double-edged sword because the experiment worked. So you saw Parkinson's patients get out of their bed and their symptoms were gone. And then about in the same year, George Coetzee, another pivotal player, was able to improve on Hornikovich's original idea and come up with L-DOPA therapy. Well, now the climate of Parkinson's disease is completely different. For one, we know a lot more about the disease. We know a lot of people have it, and a lot of people are living with it. 10 million people worldwide are living with the disease, and in the US alone, about 50,000 are diagnosed every year. So since there are so many people that have the disease, there's a ton of money being pumped into Parkinson's disease. And it's a really big thing. But even with all the money and the research that's going in, the best treatment we have right now is L-DOPA. And even then, L-DOPA isn't a long-lasting treatment. It's really just short-term and symptomatic. So, but one thing that research has been able to pinpoint is what causes Parkinson's disease. So Parkinson's disease is caused by the loss of dopamine neurons in the substantia nigra, which is a region in your brain that helps coordinate movement. So if you, so this is a picture of an unstained substantia nigra, and the black pigment is from the pigment of the dopamine neurons. But you can see, in Parkinson's disease patients, the dopamine's just not there. It's missing. It's gone. And so we know that this is the cause of Parkinson's disease, but we don't really know why this dopamine cell loss occurs. In reality, it's probably a combination of a ton of factors. Genetics is one of them. So a bunch of genes have been linked to Parkinson's disease. One in particular is the DJ1 gene. So defects in the DJ1 gene has been known to cause severe forms of Parkinson's disease, early onset severe forms. And in research, we found that if you boost up this gene, if you upregulate it and enhance it, it's been shown to have neuroprotective effects against Parkinson's. So it seems like we found a miracle gene that Now the question is finding a way to upregulate this miracle gene that can then be used to protect against Parkinson's. So, phenylbutyrate is FDA approved. It's on the market, and it's been on the market for about 30 years to treat urea cycle disorders. And in 2006, the lab that I'm working in, found out that phenylbutyrate upregulates this DJ1 gene, so it boosts up our miracle gene. And in 2011, phenylbutyrate, they found that, the same lab that I was working in, found out that phenylbutyrate stops the progression of Parkinson's disease in mice. So it's been so promising, and it's been working so well that it's now in clinical trials. So yeah, it seems like we found this miracle drug that boosts up our miracle gene, and we've cured Parkinson's, and that's the end of the presentation. Except, not really, because the mechanism of how this drug works is still really unclear. We know that the drug works, we've seen it happen, we've seen it upregulate this gene, we've seen it protect against Parkinson's disease, but we don't really know how. So the running hypothesis is that phenylbutyrate in the body gets broken down to phenylacetate and acetate. Phenylacetate and then acetate. And this acetate is what's going around and acetylating things and activating things and turning on stuff. So normally, so this is a picture of the DJ1 gene, and this is the promoter region of the gene. So normally, the promoter region is what initiates the rest of gene transcription, which then, gene translation, and that's how you make proteins. So the promoter region, there's a ton of things that hang out in the promoter region, and one of those things, and what I'm looking at, is the SP1 transcription factor. So normally, the SP1 transcription factor binds to the promoter region, and that initiates DJ1 gene transcription, gene translation, proteins. And so the hypothesis is that phenylbutyrate, when you introduce phenylbutyrate to the cell, that acetate that I was talking about, that little group, that is going to go on to acetylate or activate this SP1 transcription factor, which then binds even more tightly to the promoter region, which then increases DJ1 transcription. So if this is the case, if you introduce phenylbutyrate to a cell, you should see an increase in SP1 acetylation. So last summer, that's exactly what I did. I looked at SP1 acetylation in response to phenylbutyrate, and it was actually pretty successful. Like, I got some pretty good results. So if you look here, this is the control, so like no phenylbutyrate. The cells have not been introduced to phenylbutyrate. And then once you put phenylbutyrate in the cells, you see a spike in SP1 activation. And when I looked at it statistically, it was pretty significant. So when I first looked at this data, I was like, this is it. This is the mechanism. This is it. That's the project. I've cured Parkinson's. Like, this is the mechanism. This is it, guys. But actually, it's a lot more complicated than that. So if you look at, so in the original hypothesis, I said that phenylbutyrate will just activate the SP1 transcription factor. So if that was the case, if you increase concentrations of phenylbutyrate, step by step by step, you should see SP1 activation increasing step by step by step, but you don't really see that. And in literature, we found that 150 to 300 micromolars is the most effective concentrations of phenylbutyrate to upregulate this gene, but like, I didn't see that there. So this could be just human error, and it could just be me doing science and it not working, but I took it a step further anyways. And so I looked at this and I thought maybe there's like an intermediate step from here to here. So I revised my hypothesis. And I introduced this protein called TAF1. So TAF1 is a transcription factor unit with nuclear acetyltransferase activity. So, acetyltransferase. So I looked at it and I thought maybe it's helping, transfer this acetyl group. So, so the revised hypothesis is that phenylbutyrate, when you introduce it to the cell, it interacts with this TAF1 acetyltransferase, and then TAF1, is transferring the acetyl group over to the SP1 transcription factor, and that's maybe the mechanism. So I came up with this hypothesis because in other published literature, TAF1 has been known to assist SP1 acetylation, and TAF1 has been known to increase SP1 binding to the promoter. But no one's actually looked at phenylbutyrate and DJ1 specifically, so that's what I did. So the best way to determine if something's crucial in a mechanism is to completely take it out. So what I did, or tried to do, was, so normally, if phenylbutyrate, is working with TAF1, you would see DJ1 transcription. But if you remove TAF1 completely, ideally, if TAF1 really is crucial in the mechanism, you should see no DJ1 upregulation. And it have no effect. So to do that, I worked with temperature-sensitive TAF1 mutants. So these mutants, at low temperatures, at 33.3 degrees Celsius, they work normally and TAF1 activity is there. And then at high temperatures, TAF1 activity is completely gone. So it shuts off TAF1 activity. So it's just a way to just regulate the TAF1. And the running hypothesis then became, when TAF1 activity is normal at 33.3 degrees, phenylbutyrate increases DJ1 activity, similar to previous experiments. And then when TAF1 is shut off, phenylbutyrate has no effect. And so in theory, it was really straightforward and really simple, but then when you do science, science doesn't always come out the way you want it to. So when I quantified the data, the results were statistically inconclusive. This is my first time working with these TAF1 mutants. Before, I was working with dopamine neurons that I was really comfortable with. Like, they stained well, I could plot them, I knew what they were doing, but these new cells were just a whole new world on their own. But, I don't know, if you look at this Western blot, and you close one eye and you squint a little, you can kind of see there's something going on. So let me walk you through that. So, wild type. So this is a Western blot showing you DJ1 in response to phenylbutyrate at 39.5 degrees. So remember, at 39.5 degrees, TAF1 activity in mutants are shut off. So in the wild type, TAF1 activity should still be functioning fine. So without phenylbutyrate, DJ1 isn't upregulated. But then when you introduce phenylbutyrate, TAF1 activity should increase in wild types, but in mutants, it should still, nothing changes. So if you kind of look at this Western blot, and like I said, shut one eye, squint a little, imagine. You can kind of see that this blot is a little bit darker than all the other plots, which tells me something's going on there. To me, maybe I'm a little biased, but I think that there's something going on. I think there's definitely a trend. But science is science, and when I looked at it statistically, it wasn't quite there. But we are working on that. Specifically, I am working on that. So, a lot of people are wondering, why am I still doing research on a drug that's already in clinical trials? So, contrary to what a lot of people think, and a common misconception that I originally had, stages of research it's not cells to animals to humans, and it's not linear one way all the time. In reality, you could be doing animal research and then doing cellular bench work at the same time, or doing human research while doing cellular research. And you do it because if you find something at your bench, doing cellular work, and it could potentially improve your drug, or if it gives you more insight to what side effects there are, that's actually really important. So in my case, phenylbutyrate, we're giving this drug to humans, but we don't quite know what the mechanism is. So if I figure it out, I might be more insightful to what the side effects are going to be. And for how notorious the FDA is for being nitpicky, they don't actually care about the mechanism of the drug. All they care about is if the drug's toxic and if it's effective. So does your drug kill someone? Does your drug kind of kill someone? And if it's somewhat equivalent to a drug that's already on the market. And so if the FDA is only caring about two things, then clinical trials should be pretty smooth, right? Right? No. I don't know why I keep trying to use So clinical trials is actually pretty extensive. And now clinical trials is after you've done like, your cellular research and your animal research. Clinical trials is 3 phases. Phase 1, where you're looking at safety and tolerability. Phase 2, where you're looking at efficacy. So you're doing like double-blind trials, you're doing dose-response trials, and phase 3, which is long-term, large participation pools, and it can take anywhere up to a decade. And as you're going through from phase to phase to phase, you're pumping in more time and more money. Each phase is longer than the previous phase, and it takes more money than the previous phase. So just to paint a picture of where phenylbutyrate's at, we started this drug research in 2006, and it's now entering phase 2 clinical trials. So phase 2 is, ideally, it's a year-long experiment where you're testing the maximum dose of the drug, maximum FDA-approved dose of the drug, and then you're doing 2/3 maximum dose, 1/3, and then control, placebo, no drug at all. And you want 25 participants per group at the end of your trial. But you also know that it's a year-long trial, so not everybody's going to stay in the trial the whole time, so you over-recruit. Instead of finding 25 people per group and finding 100 people in total, you try to find 200 to try to compensate for all the people that are going to drop out. Because if you're going to pump that much money into like a clinical trial, you want your results to be significant at the very end. Like if you have a group of like 2 people, you're not going to find anything. So you over-recruit, and it's not like research methods where you just post a sign-up sheet and everyone just goes and signs up. You actually, yeah, shocker. But you actually have to go and recruit them. So you have to fly out to your patients, talk to them, convince them to join your study, and then pay for their costs. So it's a lot of money, and it's complicated. So ideally, even though the actual research portion is a year long, once you get participants, you start like the rolling applicant process. You start them right away, as soon as possible, and then hopefully, ideally, if everything goes well, it's going to end up being a 5-year experiment, even though the actual testing process is a year long. So, yep. So here we have like a really promising drug, but it might not reach the market for another decade. So, just in summary of my talk, phenylbutyrate increases this DJ1 gene, and we've seen that phenylbutyrate increases activated acylated SP1, which then binds to the DJ1 promoter and initiates DJ1 production. TAF1 may be involved in SP1 acetylation, but we're not totally sure. Like the results were inconclusive. And drug research is very tricky, and drug regulation is even more tricky. Yeah. So in conclusion, phenylbutyrate may be a future treatment for Parkinson's disease. And if you see it on the market, 10 years from now, 15 years from now, don't forget about this presentation. I was here, I'm in the background trying to figure out how the heck this drug works. So that's me. Just a quick shout out to Dr. Bashan for being a super awesome advisor, and then Dr. Fried for adopting me into your lab. Dr. Franco for being my reader, Dr. Howard, Dr. Kleiner for just kicking my butt, honestly, for trying to get me to write this thesis. And then the members of the Fried lab, especially Christopher Chow for doing a lot of the dirty work for me, and then Brianna Sims for showing me everything that I needed to know. And then lastly, the GEMS program that funded this research at the very beginning in 2015. And thank you to you guys for coming and listening. That's it.
Well, thank you, Lynn. We do have time for questions and comments and discussion. So,
Well, I will start, and that's traditionally the advisor starts. You and I spent a lot of time squinting with one eye at your TAF1.
Want me to bring it back up?
Yeah, yeah, you can bring it back up if you want a little visual for this. And I'm convinced, as convinced as you can be by one Western blot that there might be something there. But I wonder if you could talk about if it's not TAF1, if it turns out that TAF1 isn't the intermediary, Are there other candidates? What are some of the other candidates that could be between phenylbutyrate and SP1? And then also sort of related question, why did you start with TAF1?
Okay, so I'll start with that question first.
Okay.
So I picked TAF1 because I looked at the original data and it just seemed, okay, so transcription factors, when they bind to promoters, they're not, I oversimplified it, so I called it the SP1 transcription factor. But there's actually a ton of other transcription factors that it interacts with, and a bunch of things bind to the promoter. So one thing that's part of a transcription factor is like the transcription factor subunits, and then things that help acetylate things. So I was essentially doing research on what acetyltransferases there were to help, move acetyl groups from one end to the other. And the other reason why I picked TAF1 is that some of the background research that I did, at first when I looked at SP1 transcription factor, I looked at it in the whole cell, and I found out SP1 transcription factor just isn't, it's not in the whole cell, it's not in the cytoplasm. So I switched gears and I looked at it at SP1 in the nucleus, just the nucleus. I would isolate just the nucleus and try to quantify SP1 there. And I found out that there was a ton of SP1 in the nucleus. So when I was researching for acetyltransferases, I specifically wanted nuclear acetyltransferases. And TAF1, at least in literature, was the only nuclear acetyltransferase that has been linked to the SP1 transcription factor. So that's why I chose that experiment. And the other reason why I chose TAF1 is because the lab that shipped me those cells already made these mutants that were temperature sensitive, and that turned off TAF1 activity. So it was like, that was super easy, I'm gonna do that, and that's how it's gonna be. So, long answer to that. Yeah. And your other question was, if it's not TAF1, what could it be? I'm not sure, because the SP1 promoter is actually on a variety of genes, but when we tested phenylbutyrate, like when we screened phenylbutyrate to see if it upregulated all the other genes, we didn't see any other genes being upregulated. It was just this DJ1 gene. So we're not sure. Like I guess my long answer is like, if we're not sure, we know that it works. We know that there's something correlated there. I haven't specifically looked at other things since I've been trying to narrow my focus. So.
Well, and so then I kind of have another related question, which is, what's the next experiment that needs to be done? And if you had, and I'm not advocating that you postpone graduation, but if you had another semester or two in Dr. Fried's lab, what's the next experiment that you would do?
Right now, my #1 priority is trying to repeat this experiment and trying to find a way to, so before, like I said, I worked with dopamine neurons, and then I switched over completely to these temperature sensitive cells, and these are hamster cells. They're not dopamine neurons for sure, so like, they're a little bit different. They work the same way, that they turn on, they turn off, but just to look at TAF1, I switched over completely. So I think, ideally, theoretically, if I could find a way to regulate TAF1 in dopamine neurons, like the cells that I know and love and like have been working with for 2 years, then I would. But as far as I know, nothing's been done, and CRISPR is hard, so I'm not going to try this.
Oh, CRISPR's
No,
not that
CRISPR is
far
easy.
CRISPR
Much easier than that.
well, maybe, but yeah, ideally, looking at dopamine, looking at TAF1 and dopamine neurons.
Those are the only questions I have for right now.
Well, I have a question related to the first one Max asked. First of all, do you know the fold difference between your beta-actin and your DJ1?
I'm sorry, what was it?
The fold difference in expression, because that's why you have your beta-actin fold. This is your loading
Yes.
control.
Yes.
So do you know how much of an increase do you have in your DJ1? Because you say you can squint and see, but I want the numbers, because that's where your beta-actin is there.
Right.
Right?
So I did quantify it. I
Okay.
did look at it statistically and across. So this is just cells at 39.5 degrees. So I also have like the 33.3 degrees and the base at 37 degrees. So I looked at it, and when I quantified it, it wasn't significant, even with the beta-actin control.
But forget about significance. Did you see an increase when you quantified or not?
Not really. It was just all over the place. Like it was just all over the place.
Okay. And my second question is, how do you know that phenylbutyrate is specific to acetylating those particular genes? Because if it acetylates a bunch of genes, then we know that acetylation increases gene expression in general, then you may be just lucky to have this gene overexpressed.
Yeah. So we don't know if phenylbutyrate is specific yet. But like I said before, we screened a bunch of genes, and the only gene that we saw increase was the DJ1 gene. So maybe it's not even acetylation at all. Maybe it's something completely different. But as far as I know, it's complicated because before me, way before me, there was a guy that was looking at HDAC inhibitors, which is the kind of drug that phenylbutyrate is. He was testing a bunch of HDAC inhibitors to see which one best upregulated the DJ1 gene. And for some reason, phenylbutyrate and butyrate was. So if you look at the structure, something about the phenyl group, I don't know, makes it better at upregulating this DJ1 gene. And I think there was another study where some guy looked at phenylbutyrate and then phenyl acetate, longer chains to see if the longer chains of fatty acids will upregulate this gene even more. And it turns out no. So more acetyl groups, no. Less acetyl groups, no. It's just this phenylbutyrate for some odd reason. So we don't really know. That's kind of why I did this, but it's
Thanks.
yeah. Sorry.
No, that's good, and that's impressive work.
Yes.
I'm interested in you said that this drug is already being used to treat a kidney condition?
Yeah, in Rio 7 as well.
So is this something that if you give people, are you expecting there to be some kind of side effects that would be linked to the kidney, or can you take this so that it is specific to the brain?
So far, the published side effects, as far as the FDA, what the FDA has written out, has mostly been GI, so nausea, vomiting, diarrhea, stuff like that for the drug. We do not, I do not know the long-term side effects of it yet, because I guess that's why we're doing the clinical trials. But as far as kidney effects, I'm not totally sure. I know there was a girl in the lab that was looking at urea in response to phenylbutyrate, and I can't remember what she ended up finding.
And is this a drug that's taken orally and it crosses the blood-brain barrier and all that kind of stuff? They know that?
I know that you take it orally. I'm not totally sure if it's. Yes.
That was really wonderful, Lynn. Thank you. I want to ask you a question. So you're clearly a scientist, right? An accomplished scientist, right? But you also have other interests, right? You're religious studies minor, and you're really good sort of in our honors seminars, right? And you're a reader and all that kind of stuff. So I'm curious, and knowing you, I know that these things are not separated, right? There's a way in which you sort of try to live in these worlds with some kind of integrity. So I'm wondering, are there some ways you think sort of being a student of religion has informed you and sort of made you a better scientist, and then also being a scientist has sort of somehow informed the work you do in the humanities and made you better at that? And can you speak to that at all, or say something?
Yes and yes. Short answer. So right now, I'm actually taking this class called Theology in the Context of Science, and it's basically a class about how you pretty much interpret your religious background, your Catholic background, and incorporate all these different science lifestyle whole. And I would say that my religion has made me more passionate, more empathetic to why I'm doing the research. I can't exactly say that I'm always thrilled to be at the bench all the time, but I am thrilled about the outcome of my research. I'm thrilled about helping people, and I'm thrilled about all the people that it might affect if this is successful. So yes, on religion. And in reverse to that, being a scientist has made me kind of question a lot about religion, kind of address it and have a conversation with it. So yes.
So when I read a very early draft of your paper, I came away from it thinking that you were very pessimistic about the FDA and the FDA process. And as I've read more and more recent drafts, I feel like I've detected a sort of change in that you've taken out some of that sort of criticism and pessimism. And I'm wondering if you could sort of tell me where you are now. Are you as pessimistic? Are you pessimistic about the FDA process? Are you not pessimistic? I can't tell where you are now.
Okay. So you're right. When I first started doing this research, I was like, the FDA stinks. They take way too long to approve drugs, and nothing's going to come out of it. But the more research that I did, it was like there is more research on, or there are more published articles about how helpful the FDA is than how much it harms. And I think it's just like reading about all the things that went wrong in the past has kind of like, oh yeah, I guess like we do really need this. And I guess this is a thing. Like we should pay attention to it. And like drugs, it's really hard to test drugs to test long-term side effects because when you first take a drug, like it could be great and it might turn on some genes, might turn off some genes. But when you take a drug over long periods of time, how your body reacts to the drug changes. And so I guess like I was super impatient at first because it, like we're not moving any drugs into the market, but like if you really want to make sure that it's safe, it takes some time. So I've changed a little bit, but I'm still kind of like there needs to be FDA reform. I'm just not sure how it looks. So. Yes.
Well, I have a pretty softball question. So I remember back in the day, there were a couple times you coming into my office, and you're doing this science and honors in neuro and your honors thesis as well. What advice would you give to you when you were first coming into my office? So what advice should I be giving to the juniors who are embarking on this process?
I can be really cheesy and say go with something you really like, because I definitely did. But more realistically, I think, for me at least, I think I tried to go too broad at first. I tried to find something that my scientist side was like, yes, and then my religious side was like, yes, and you can't do that all at once. So like for a while there, I was kind of swimming, and I didn't know how I wanted to narrow it. But then picking a topic that's narrow enough that you can continue to learn from, and then just sticking with it. This is the topic. This is it. I'm going to take it all the way.
So I just want to say how great it was to have Lynn in the lab and working hard and really committed to this project. And to meet your extended family here.
I have a question from an early slide. You talk about this drug being effective or bringing results for people with early onset Parkinson's or severe, right? And so I guess I'm wondering, and then you kind of seem to go to say this would be effective for all Parkinson's, or it has promise for all Parkinson's patients. Is there a distinction between types of Parkinson's, or how it develops, or when it develops? Right.
So right now, the best way to diagnose Parkinson's disease is symptomatically. So if you look at a person and you see he has like resting tremor and all that stuff. So it's really hard to say this disease can just prevent Parkinson's, because like it's a fatty acid, so you don't want to take it up way too early and you get like all these digestive side effects. Like that's not a good idea. But I would say how this drug works is upregulating stuff on the genetic level. So once you get diagnosed with Parkinson's, I guess yeah, you can take it. But also during that point, there's L-DOPA therapy and all these other things that you could try first. And for me, since those are FDA approved, they've been around for a while, I would personally start that first and then go to phenylbutyrate, at least until we know more about it.
Thank you, and just sort of congratulate. You're all invited to join us tomorrow, I think, 2:30. Brittany tomorrow?